Thanks for the invitation to speak in this forum. I'm really looking forward to chatting with more of you and nice to meet you virtually. So yes, my name is Sabrina and my lab is just starting its second year at the University of Arizona. We are focused on disease ecology, and in particular, my interests lie in how animals respond to emerging parasites and pathogens. So I'll be telling you about some of the work that we do. And let's see if we can make this work. So I'm going to be focused on islands, on the Galapagos in particular, and I love islands because they're excellent natural laboratories for studying emerging disease. They tend to be isolated, fairly closed systems, and their communities are also threatened for a number of reasons. We estimate that humans may have driven more than 1,000 bird species extinct in the Pacific, and disease certainly played a role in that. So I do, in fact, work in the Galapagos, which is this little volcanic archipelago off of the coast of mainland South America. They were first made famous by Darwin, but have also become well known in the years since for some historic studies on evolutionary biology by Peter and Rosemary Grant. And they're special for an additional reason, and that's because they've only been inhabited by humans for a relatively short amount of time, at least compared to other islands in the Pacific. And so that means that the disturbance that humans cause has been relatively recent, and that gives us a great opportunity to study this disturbance in contemporary time. So a lot of people, when they think of the Galapagos, they might imagine something that looks like this, absolutely beautiful, pristine, untouched. And that's true for a lot of the archipelago, but a lot of the islands also look like this now. And so this is the main town where I'm based, Puerto Ayora. And the rise in tourism and the development of the Galapagos over the last few decades has really introduced a lot of ecological change. And principal among that include invasive species, our favorites, rats and cats, as well as invasive plants and introduced parasites and pathogens. And the main focus of my research has to do with how these environmental changes affect Galapagos birds, including Darwin's finches, one of which is shown here. And today I'll talk a little bit about a virus, since I think you all care about viruses. And that is avian pox, a virus in the poxvirus family, the same family that caused smallpox in people, as well as mpox, very diverse group of DNA viruses. In birds, these viruses are found all over the world in lots of different bird families, but from specimen records, we estimate that it has been present in the Galapagos for about 100 years. Infection causes these lesions on the feet and faces of the birds, and many different passerines in the Galapagos, these songbirds, are susceptible to infection. And so one project that I'll talk about briefly had to do with the transcriptomic responses of Darwin's finches to pox infection. And this was kind of an initial foray that I did into the system as a postdoc at Cornell University. And I basically wanted to know what genes are activated in response to infection, and what are the functions of those genes? And I thought by answering these questions, we could learn a little bit about how these relatively naive hosts respond to this emerging pathogen, and whether the response might vary among different host groups, whether it's adaptive or not, that sort of thing. So this project began in the field. We can catch birds using these mist nets, take blood samples from them, look for signs of infection, and I'll highlight the contributions of two principal members of my team, Hanai and Diana, who were really involved in this work. Then with these samples back in the United States, I designed an RNA-seq experiment with the help of our core facility at Cornell. I focused on two focal species that are both found in the same place, same community, the medium ground finch and the vegetarian finch. These are both endemic to the Galapagos and descendants of a common ancestor, but they're from different kind of parts of the Darwin's finch clade, so they're not sister species. And we sequenced 10 infected and 10 uninfected individuals. That produced a lot of mRNA reads with pretty good alignment to our reference genome, giving us information of for the expression of about 16,000 genes. And then I analyzed the potential biological relationships and functions of those genes using something called gene set enrichment analysis. So just to jump into some results, the main message was that infected birds of both species upregulated expression. So looking at infected versus uninfected ground finches, for instance, our results provided a set of some differentially expressed genes. So in this volcano plot, the x-axis tells us the difference in expression between infected and uninfected birds, and the y-axis tells us something about how confident we are that that difference in expression between groups is significant. And those points that are colored and labeled are genes that we identified as significantly differentially expressed in infected birds. But then we can also take the expression information from all of these genes and do that gene set enrichment analysis that I indicated, and that produced something that I'll call significant hallmark sets. And so these are sets of genes that we know to be biologically related and to be involved in certain particular biological processes, such as the inflammatory response. And so when we identified significant hallmark sets, that told us that genes involved in something like inflammation were significantly differentially expressed between these groups. So to just expand on this GSEA result, again, comparing infected to uninfected ground finches, we identified a number of significant hallmark sets, and there's plenty that we could talk about here. I know there's a lot of immune-focused words, but just to kind of summarize some of what we saw, a lot of these hallmark sets have functions in innate immunity. And so that's everything from things like interferons, like interferon alpha, which is one of our first lines of defense against viral infection, to immune signaling, to general immune processes like complement and inflammation, and even things like programmed cell death, which we also know to be involved in an antiviral response. And then beyond these sets, we also saw a number of significant hallmark sets that seem to have some function with tissue repair. So that might be coagulation, for instance. And tissue repair might also make sense in that the infection causes these lesions on the bird's feet and face that they presumably want to repair. And so in sum, a lot of these results were sort of reassuring because they indicated that these birds are responding to a relatively new virus in ways that we would expect a vertebrate to try and fight off a viral infection. And that means also that because a lot of these processes are well known from human studies and studies of other model organisms in response to poxviral infection, these results really underscore the conservation of that antiviral response in invertebrates. But we also saw some really interesting differences between species. So looking at the vegetarian finch, infected versus uninfected groups, we did see some of the same immune sets that were significant in our GSEA analysis. However, the p-value support for these differences was somewhat lower. We also saw some of those sets that we are associating with tissue repair. But there were a number of sets that were kind of strange, had something to do with cell function, with cell metabolism. And looking at those ones, we were starting to see signs that maybe suggested that the virus is manipulating the host in ways that would change gene expression of the birds. And just to give you a quick example of why we think that, this cholesterol homeostasis set was significantly different between infected and uninfected vegetarian finches. And at first blush, I didn't really see a connection between cholesterol and host virus interactions. But digging into the literature, I realized that viruses actually care quite a bit about their lipid environment, and that's for a number of reasons, including that they have to pass through a lipid membrane to gain entry to the cell, that they need lipids in order to replicate and make those virus building blocks. And so other studies in laboratory settings have shown that poxviruses, their replication can be limited when cholesterol is limited. And that suggests that these transcriptomic changes that we're seeing in the host that have to do with cholesterol homeostasis, they could be a signal that the virus is really gaining a foothold in this host and is really able to co-opt the cell's machinery to its own ends. And so, of course, we don't know that for sure, but I thought that this broad picture look at all of these gene expression changes in these hosts can provide us both clues into the host's defense and also into this interplay between the virus and the virus's interest in controlling the host cellular machinery. So we're following up on this project with continued monitoring and studying of this system on Santa Cruz Island in the Galapagos. And I'm conducting this work with a really wonderful team of Ecuadorian and international researchers. And our future projects include studying how disease resistance and susceptibility varies over this Darwin's finch tree. We know Darwin's finches are a famous group in evolutionary biology. There's some 18 different species. And this radiation provides really fantastic replication to study how repeatable or not host parasite evolution is when hosts encounter novel viruses. The current leader of this project is my technician, Deanna, in the Galapagos. So before I end, I just wanted to say a couple of things about community education and work that I do on the islands. I recognize that as a foreigner, I'm really privileged to get to work in this area. And I want to make sure that I'm contributing to the development of the community there as well and giving something back. So I've been conducting community education in various ways in partnership with the Charles Darwin Foundation for several years. Our general principles that guide these efforts include improving access to science education, building local capacity, and supporting those local management and conservation efforts. And so they include targeted training for people who are working in biology on the islands, as well as kind of general workshops on bird diversity, conservation, et cetera. One of our workshop groups is shown here. And these are just some of the themes that my lab works on that I don't have time to talk about today, but would love to chat more if any of you work on these things. But in the interest of time, I'll just say thanks to my funders and my collaborators, and thanks to you all.